Welcome to this biochemistry lecture on nucleotide biosynthesis. Just as a general review of nucleic acids, there are two types of nucleic acid. There are the purines and the pyrimidines, and you can see those in the center there in blue. The structure of a nucleic acid, some of the terms associated with it. The nucleoside is the base plus the ribose, and in RNA, we have a hydroxyl group at the 2' position, and a hydrogen in DNA at the 2' position. If we add in the phosphate groups, you can see that we, via phosphodiester bonds, you can see that we are going to be at a nucleoside, monophosphate, diphosphate, triphosphate. So the nucleotide is composed of three components: the base, which is in, if you look at the far right figure, is in light blue, the ribose sugar, which is in black, and the phosphate groups, which are alpha, beta, and gamma labeled, and they are in purple. So that's a general overview of nucleic acids, which is important for manufacturing, obviously. So here are the five nucleotides. That there are the two major purines, adenine and guanine, and their structures. And then we have cytosine and thymine, which are DNA. And then we have uracil, which is for RNA. Cytosine is also for RNA as well. But these are the major nucleotides. All right, there are two ways that we can manufacture or recover the nucleotides. We have the de novo pathway, where we create and manufacture a nucleotide from scratch, and the salvage pathway, which is where we take and we recover those nucleotides that have already been manufactured. And there's not really a difference between the two, and both of them are used pretty interchangeably. All right, so there's, again, purines and pyrimidines are manufactured differently, and if we're going to go de novo, purines start with PRPP, which we'll talk about here in a little bit, and they go 11 steps, and we end up with ATP and GTP at the end, whereas with pyrimidines, we have a simple 4-step metabolic process to create the pyrimidine ring, and from there, we are going to get to UTP. From UTP, it's a simple reaction to get to CTP. And these are all going to be RNA, and then if we want to go to DNA, there's a simple enzymatic reaction to replace that hydroxyl group with a hydrogen. So that's the general theory of what we're going to discuss today and how they fit into the big picture. Let's talk about the de novo pathway for pyrimidines. We start with bicarbonate and ammonia. And basically an amine group. Takes 2 ATPs. We're going to go to carbamoyl phosphate. We're then going to create a pyrimidine ring by taking carbamoyl phosphate and a aspartate, and then we're going to take it down to UTP. An important consideration, and that you're going to want to know for exams you take on this topic, are where the different elements of the ring come from. So you might notice the C2 and the nitrogen at the position 3 come from the carbamoyl phosphate, and the nitrogen at 1 and the 4, 5, 6 carbons all come from aspartate. You'll want to know that for sure for the exams. So let's get in a little bit more detailed on this pyrimidine biosynthesis pathway. So again, we start with bicarbonate and an amine. And in an ATP catalyzed reaction, we are going to create carboxyphosphate. And that's the first step. The reaction is catalyzed by carbamoyl phosphate synthetase, which you see on the right. There's multiple active sites, and it's really cool to note that there is a channel that goes to the middle of this molecule, and there are multiple enzymatic sites that occur within this reaction. So we're going to go from bicarbonate to carboxyphosphate to carbamic acid, and then from carbamic acid we're going to go to carbamoyl phosphate. Along the way, we end up adding a phosphate in the first group. we end up adding an amine in the second group, and we end up adding another high-energy phosphate group in the carbonoyl phosphate, all the way through the inner channel of this. Carbonoyl phosphate synthetase, CPS, is involved in this, and that is a multiple step reaction that occurs. The next step is we go from carbonoyl phosphate, and we're going to add, with an aspartate group, we're going to go to carbonoyl aspartate. That's going to create the almost ring structure. From there, we go to dihydroorotate, which we complete the ring and in a hydrolysis reaction. And in the last step, we go from NAD+ to NADH+, and we're going to create a double bond on the orotate. That is done by the ATCase, which is allosterically regulated. CTP inhibits the reaction, and ATP stimulates that reaction. It's also controlled by gene regulation. This is one of the classic examples of allosteric inhibition and gene regulation that you will find that are associated with enzymes. People often use this in those reactions. Okay, so the next step involves PRPP, which we'll talk about right now. PRPP is important as one of the steps to the next. So we're going to go from ribose 5-phosphate, and PRPP synthetase is going to enzymatically catalyze that reaction to get to PRPP, which is important in the next step. So we're going to merge PRPP and orotate into forming the pyrimidine structure, and that is where we're going to start to get towards the end of nucleotide biosynthesis. So from here, we go to orotate with PRPP, and we get to orotidylate. Orotidylate is then decarboxylated to form uridylate, which is also known as a precursor to UMP. And basically then we've formed the pyrimidine structure, which you see in that reaction. So we've got, that's a nucleotide. You can see we got a high energy phosphate on that carbon, and we've got our pyrimidine ring that's attached to the ribose structure. So from there, we've got basically what we need. And so PRPP provides the ribose ring, the orotate provides the pyrimidine structure, and now all of a sudden we're at the manufacturing of nucleotides. From there, we can manufacture CTP with by transaminating, by taking off the amine group from a glutamine and add it instead of that carboxyl group to form CTP from UTP. And that's a simple reaction. And so the other pathway that we can utilize is a salvage pathway, where we're going to use preformed pyrimidine bases, and then we're going to recover them from nucleic acid breakdown. And then we're going to use those to synthesize new nucleotides that are going to be incorporated into DNP. The example here we have is thymine, which is a part of DNA degradation, is salvaged, and it's going to be incorporated into a nucleoside by thymidine phosphorylase. That's the recycling pathway that occurs. And from there, thymidine kinase will generate the nucleotide. So that's the salvage pathway that also can be used to generate pyrimidine nucleotides. The next example is purine biosynthesis. We're going to talk first about the de novo pathway. The purines are synthesized in a 11-step reaction. There's a 9-step reaction that occurs, and then to get to a final nucleotide, there's a 2-step one. There's a 9-step generic one that applies to both nucleotides, and there's a 2-step process that occurs for a specific one. And the general idea is we're going to take PRPP and we're going to generate the molecule nucleotide from that. All right. This is an important overview slide where we talk about after we have a purine ring, it shows where the different molecules come from, and you can see that from every step of the reaction. We have the nitrogen, the N1 comes from aspartate, the C6 comes from CO2, the C5 The C4, 5, and nitrogen 7 come from glycine. The C2 comes from the formamyl tetrahydrofolate. The N3 comes from glutamine. Know where those come from because that's going to be an important part of your exam. So from here we're going to have a purine formed. We're going to go to IMP, and from IMP we can separate into the two RNA, ATP and GTP, and then from the ATP to GTP, the ATP to DNA ATP, there's a simple conversion, a dehydroxylation reaction that occurs for both when we convert from RNA to DNA. That's the general theory of the pathway. So we're going to talk about the top process, then we'll talk about salvage. This kind of gives you an idea of how we get the last steps from IMP to the specific nucleotide. So in purine biosynthesis, is a 9-step reaction. Generic, the first 9 steps, they're an analogous reaction. The first six steps are analogous reactions. Each of the steps consists of an activation of an oxygen by phosphorylation, and then we're going to displace that phosphate with an ammonia or an amine that is going to act as a nucleophile. So that's the general reaction steps that's going to occur in the first 6 steps of this process. We're going to activate, and then we're going to have a nucleophilic attack on that. In step 1, glycine is going to be coupled to the amino group of the phosphoribosylamine. Second step, we're going to take N10 formyl tetrahydrofolate, which is abbreviated THF, and we're going to transfer a formyl group to the amino group of the glycine residue. So we're basically starting to build up this reaction. Step 3, the amide group is phosphorylated and converted into an amidine by the addition of an ammonia derived from a glutamine that is going to be part of the reaction. So in that step, in step 3, we end up with the formyl glycinamidine ribonucleotide. Apologize for the spelling, the pronunciation. We almost have a ring structure at this point. In step 4, we're going to do an intermolecular coupling reaction that's going to form that 5-member imidazole ring. And so we're going to add in and basically form the imidazole ring. So we have a 5-amino imidazole ribonucleotide that's formed at this step here at the end of 4. Bicarbonate then adds in the first exocyclic amino group. You can see that in purple. And then a carbon atom is going to be added. From there, onto that amine group, the imidazole carboxylate is then going to be phosphorylated in step 6, and then it's going to be displaced by the amino group of aspartate, from aspartate. So end up going from carboxy amino imidazole ribonucleotide into step 6. The product is 5-amino imidazole 4 N-succinyl carboxamide ribonucleotide. That's a mouthful there. We're then going to take off and chop off the fumarate ring, or the fumarate functional group in step 7. Then we're going to add a second formal group that's going to this, or a second formal group is going to donate. It's going to be donated to the THF, and then we're going to cyclize that reaction in step 9. So we end up with isocyanate, or IMP. IMP is the precursor for both ribonucleotides, and we've gone through this 9-step generic purine biosynthesis pathway. We can also salvage enzymes, or the nucleotides again, and there are 2 enzymes that are associated with that. We have adenine phosphoribosyl transferase that can catalyze the formation of adenylate, and hypoxanthine guanine phosphoribosyl transferase, which is appropriately abbreviated HGPRT, catalyzes the formation of isocyanate and guanylate. I feel like today's video is an exercise for me in correct pronunciation of complex chemical compounds. But so there's 2 reactions that are catalyzed that can recover purines from a organism. for the exam, you're going to want to know this pathways, and I think this is an important. This graphic kind of gives an important understanding of how these different nucleotides can exist amongst themselves and how they can be recycled amongst themselves. So it kind of gives an idea of the salvage pathway and how the enzymatic reactions can then be used to get to the different forms that are necessary for the different precursor sites. So this is a summary of that for the purines. Here it is for the pyrimidines, and if you know the figures in blue, it'll help with the chapter summary and the exams, because that's where we're going to focus our efforts on. That I hope you enjoyed this lecture, and we'll look forward to seeing you in the next one.